We are very privileged to have some excellent presenters with us. So we're going to start the night with Maithé Tauber, who is the Professor of Pediatrics at the University of Toulouse. She directs the Reference Centre for Prader-Willi Syndrome in France and is the chair of the Scientific Council of Prader-Willi Syndrome in France. Her research includes oxytocin in PWS. And tonight, Maithé is going to speak to us about new data in PWS and perspectives. Thank you, Maithé.
As Georgina said, I'm going to present new data, and I chose to present our new data and make a perspective with the other data. We are a very big center in Toulouse, working with the whole country in France, and we have built a network in research. And I will present research in many dimensions of Prader-Willi syndrome and the perspectives that the results led to. So first, my disclosure as ever, and then the outline, because it is a little bit tough. So I want to make outline. Normally I don't make one, but I think it's important today. Just I will talk about mental health, endocrine and metabolic troubles, scoliosis, the biomarker of scoliosis, COVID-19 issues, and therapeutic perspective. But first, giving an overview of Prader-Willi syndrome, as you all know, I'm sure that PWS is now viewed as a neurodevelopmental genetic disorder due to loss of expression of paternal inherited maternal imprinted alleles in the chromosome 15, Q11, Q13. What is new nowadays for 10 years, I must say, that the frequency of maternal disomy at birth is increasing, probably due to the increased of age of mother, but I say also of father, because we have all this data in our data bank. The critical minimal region was an important issue suggested many years ago and demonstrated in mice by deleting this critical region in mice and reproducing the PWS phenotype in mice. And then we also saw that there is a critical minimal region in human, and we had the opportunity in 2014 to publish a paper, including this young woman who display a very typical phenotype of Prader-Willi syndrome, and in whom we describe a microdeletion of the SNORD116 gene. So it was interesting for us because we start with this to develop our research focusing on the SNORD116 gene with many collaborators in France and in Europe and also in the US. So first I want to emphasize that the view that we have, I think all of us have this view as PWS as a trajectory, specific trajectory, including 3 dimensions: nutritional, endocrine and metabolic dimension, and behavioral trajectory. And of course, we all know the nutritional phases described by Jen Miller and Dan Driscoll in Florida, with this switch from anorexia and poor growth until excessive weight gain, and we can say unexplained excessive weight gain, and then switching to hyperphagia, and what I'm going to describe in the further slide of food addiction. And then we have the endocrine trajectory, which we know better, as I am a pediatric endocrinologist, so I first focus on it, but you will see that I now focus on every dimension of this very interesting syndrome. And of course, and most of importantly, I must say, the behavioral features, which are a key point of this disease, with intellectual disability, which is mild, as you know, learning disabilities, poor social skills, and emotional dysregulation. And just to finish the introduction, we now all admit that impaired development and function of the hypothalamus explain most of the typical features, including endocrine, eating disorder, behavior troubles, and of importance, the dysautonomia that we observe and that may induce very severe outcome in PWS. And we need to focus on research on this dysautonomia. And also now we are knowing more and more the roles of the genes in this region. Interestingly, the one that as I underlined in red are the genes involved in compulsive-like behavior. The one in green are the genes involved with the hyperphagia and food intake. And in blue, the endocrine features But of course, all of these genes are involved in the neurodevelopment. And I want to just pay attention to the very good review proposed by Correa da Silva last year, suggesting all the roles of the neurons, orexigenic and anorexigenic neuron, and the hyperactivation of circuits involved in food intake and motivation in Prader-Willi syndrome. And then the last presentation for the background is the knowledge that we have in the neuropeptide, that some of them are hypoactivated or hyperactivated, and also the data that we now have in the plasma concentration. So after this, I will focus on how we work on addiction, an addictive disorder and Prader-Willi syndrome. What is hyperphagia? Hyperphagia is obsession with food and food-related items, foraging and storing, stealing food, craving for food. That means taking high risk for obtaining food, food impulsivity, and before and during meals, somehow a typical behavior, which of course varies from individuals. And they also display excessive quantities of food and drinking intake, which is uncontrolled and that we saw systematically in the patients. So hyperphagia in Prader-Willi syndrome might be linked with addiction for food because all these symptoms are also seen in addictive disorders. But there are also, beside the hyperphagia, other addictions. Smoking is one of addiction that we observe. Alcohol, very rarely because it is more controlled, but also objects collections, global impulsivity, thinking rigidity, disruptive behavior, perseveration, difficulty to evaluate quantity that I already mentioned. So when we look at this with my PhD student in the last 3 years, Juliette Salle is a psychiatrist. So we make a review of the literature on how SNOD116 gene can be related to addictive disorder. And we make this observation that there is a really shared symptoms, systems, and genes between addictive disorder and Prader-Willi syndrome. And I must say, I didn't know this, but I learned that addictive disorder was characterized by lack of attachment, poor social skills, risk-taking, high reward anticipation, compulsive drug consumption, of course, but also sleep disorders and neuropeptide dysregulation. So Juliette Salle told me that we can compare, these symptoms, the systems in pair and the gene, and we came to the conclusion that possibly SNOD116 may drive some vulnerability to addictive disorder. And in the other way, I propose to think about how addictive disorder can help to care or to understand Prader-Willi syndrome. And what is the role of SNOD116? So every component of this gene, this locus, is involved in modulating some very important pathway. For instance, the OS gene, 116 OS gene, interact with RBP5, which drives the methylation of histone. The gene itself, the SNOD116, the SNOD gene, interacts and regulates the proconvertase 1 gene, impairing the maturation of the peptides, and also the long non-coding SNOD116 gene also interact with FOX2. So it really seems that this gene is possibly interesting in, of course, in Prader-Willi, but also in addictive disorder. Then we decided to work with Juliette Salle in epigenetic marks because we now know that neurodevelopmental disorders and epigenetic are very interesting and very highly linked. And we recently described a paper studying the epigenetic marks, and particularly the methylation marks in the whole genome, in blood cells, in patients with Prader-Willi syndrome, patients with microdeletion of the SNOD116 gene, and patients presenting with MAGEL2 mutations, so the Schaff-Young syndrome. And we found that more than 29,000 differently methylated cytosine, corresponding to more than 5,000 differentially methylated regions, which match with more than 2,000 genes. That means that in Prader-Willi syndrome, there is not only epigenetic marks in the region, neither in the region known to be epigenetically modified, but also in many, many genes. And what we look at is what the pathways are differentially methylated. We first underwent a cluster dendrogram that allowed us to show that there was not differences regarding age in a differentially methylated region, as we study infants and young adults, and we didn't see any differences. But what we observe is that the lowest the shortest deletion of the SNOT116 region, the closer to the control regarding the cytosine methylation. And what we observe here is that the young woman with the SNOT116 deletion is very close to the control. And now the patients bearing short deletion, type 2 deletion, are close to the microdeletion, and then the type 1 deletion, which is a large one, and then the major 2 mutation and UPD. So it was very interesting to see that this classification gave interest to the results as we observed. And then we had the possibility to study in detail the pathways that are involved in the differentially methylated region. And what we observe is that the SNOT116 gene and the major 2 gene impact on different methylated region. The SNOT116 gene is more involved in neurodevelopment system pathways and metabolic pathway, while the major 2 gene is related to macromolecule biosynthesis, which is really interesting and close to what we observe in the clinical phenotype. And then we went to studying by group the pathway of oxytocin, endocrine resistance, and neurodevelopment, and cross with the regions methylated in Prader-Willi syndrome. And we observe there is many linked with this pathway. And also obesity and addiction pathway that are methylated in PWS. You can see that there are more than 1,000 genes that are comparable in the obesity pathway and in addiction pathway. That is not a surprise. And going deeper into the gene analyzers, we observe different methylation status of each gene. Particularly here, we can see that the oxytocin gene is lower methylated than in control. And we also see here that the adenylate cyclase 3 gene, which is very now known to have a role in obesity and addiction and in diabetes, is lower methylated in Prader-Willi syndrome compared to control. So we don't know what are really the signification of this data, but we think that it's important to see that this SNOT116 gene is related to methylation to the whole genome. So we need to find what is the causes, what are the consequences, and to work more on it. So we started a program in the iPS cells to working on the epigenetic marks. Then we are going to focus on 2 points on endocrine and metabolic disease. So we published recently, last year, the role of IGF-BP7. I think it's a very new data. So we have been working many years ago without finding an explanation, and finally we did, with Sana Ediri and together with Jean-Pierre Salle, my colleague in Toulouse. So we observed that there was in the cells of Prader-Willi patients high secretion of IGF-BP7. IGF-BP7 controls the access to IGF-1 receptor. It is an IGF binding protein which has more affinity for insulin than for IGF-1, but it's really linked to the access to the IGF-1 receptor. And the first time we observed the elevated IGF-BP7 in the cells and also in the blood of patients compared to control, we want to understand what are the reasons and what could be the consequences. And then we, of course, demonstrate that when we treated with growth hormone patients with PWS as other patients, we observe an increase in IGF-1, SDS, but also we observe a decrease of IGF-BP7 levels in plasma, and they reach normal levels. So it was very interesting at this time to observe that. And interestingly, the decrease of IGF-BP7 inversely correlated with the increase of IGF-1. So we need to explain that and to understand the possible role of IGF-BP7. And what we saw is that the NOD116 deletion drives an increase in IGF-BP7. We work on the mice model with NOD116 KO, and we also demonstrate that they have the same level of IGF-BP7. And we also observe that this NOD116 induces a decrease in potential IGF-BP7 cleavage by BC1, as we already talked about. So these both actions explain why the IGF-BP7 levels in the brain and in the blood are elevated in PWS and in mouse knockout for this NOD116 gene. And when we treat it with growth hormone, we increase IGF-1, we decrease in turn mRNA for IGF-BP7, and we normalize IGF-BP7. So what could be the consequences to normalize IGF-BP7? So at this time, we don't know. But we know the IGF-BP7 is very involved nowadays. It's a very recent discovery from last year's IGF-BP7 is involved in neurodegenerative disorders. And we also know that IGF-BP7 is a marker of myeloid leukemia, which is increased in the prevalence in people with PWS. So we don't know, but what we know and what we showed is that growth hormone treatment normalizes IGF-BP7 levels, which can be of importance. We just go now to the diabetes in the transition period. I am a pediatric endocrinologist, and I was very surprised by the early occurrence of diabetes during the transition period. Diabetes occurs in 25% of patients with Prader-Willi syndrome at a mean age of 20, but we want to focus on the natural history of type 2 diabetes in patients with PWS during the transition period from 15 to 25 years. And what we observe is that the occurrence of type 2 diabetes is very early, before 20 and at a mean age of 16.8 years. We reproduce the fact that diabetes occurs when the BMI was very high. You can see here nearly 40 kilograms per meter square. But interestingly, what we found, it was not a surprise, but we have to focus on it and to use it, that these adolescents and young adults have a high parental history of type 2 diabetes and overweight. So we can do something to prevent diabetes and to screen for diabetes at this transitional period. And when we observe a very huge increase of the BMI from, you can see here, 10 years to 20, we need to do something and to prevent this increase, because we did well for children, but not as well in the transition period. So we need to control BMI in order to prevent diabetes. Because the glycemic control is very difficult to achieve in this patient, particularly in the adolescent period. You know, all the patients that we have in our study, 39, had a median of 3 anti-diabetic treatments. Why? Because it was difficult to control their behavior and then the glycemia. But there is an hope, probably something to follow, the use of sodium glucose transporter type 2 inhibitors. That, you know, there were in the literature 3 reports, and we've been using it with very, very good results. Of course, we have to careful follow it. There is not an approval for PWS, but it's something very interesting to know. Then we switch to scoliosis. What did you find regarding scoliosis in PWS? So we found that ghrelin may be a marker of the occurrence and severity of early onset scoliosis. Why did we focus on it? So first, because Prader-Willi as a group, not as an individual, has a high level of ghrelin since birth and at all ages. And secondly, because in our lab, we work on scoliosis and bone. And my colleague was very interested when I told him that scoliosis is very high in Prader-Willi syndrome. Then we start to study the role of ghrelin in scoliosis, and there are now many publications in the literature showing that ghrelin levels are associated with idiopathic scoliosis in adolescents. And then coming back to PWS, we want to know if the level of scoliosis is associated with the severity of the scoliosis, and then if the levels during the first year of life of ghrelin can predict the occurrence of early onset scoliosis in children with Prader-Willi. So what we found is, and we matched carefully the patient with early onset scoliosis. So the scoliosis started at 3.7 years. So we matched carefully them regarding BMI, which of course it's of importance with ghrelin levels, and also regarding treatment, growth hormone treatment, which also impact in the ghrelin levels. So we perform a very, I think, well-known analysis with another PhD student, Divya Pagorikona. So we saw that here in red, that children with early onset scoliosis had a lower AG UHG ratio. That means they have a sort of UHG increase. So there are more UHG than AG. And so the ratio decreased compared to the patients who did not present with early scoliosis. We also see that there is a trend for IGF-1 differences between the two groups, and also, but it is not clinically significant, earlier start of GH in the group of scoliosis, but it's only 1 month. I don't think it's of importance. And here we show, it has already been shown that possibly the patient with disomy are more susceptible to early onset scoliosis for a reason that we cannot explain in detail nowadays. But interestingly, we found a correlation between total ghrelin with unacylate ghrelin levels and the Cobb angle, which identified the severity of the scoliosis. So really, PWS as a group, high vulnerability to scoliosis. Ghrelin is involved possibly in this vulnerability. And my hope is, why not having a drug to prevent scoliosis in PWS if ghrelin level is involved? What we cannot demonstrate because of poor statistical effect is that the levels of ghrelin during the first year could not predict the onset of scoliosis. But we found here, albeit not significantly different, that children who will develop scoliosis before the 4 years of age have high level, albeit non-significant, I know, but they have higher level of ghrelin that the group who did not develop early onset scoliosis. So what to do with that? I think we have to go ahead and continue to work and to understand the role of ghrelin in scoliosis. And to finish, we published last year a survey on the COVID-19 showing that people with PWS are not at high risk for severe COVID-19 as a group, of course. So we made a survey, we discussed between us because we observed, as of course you probably, that people with PWS did very well on SARS-CoV-2 infection. So we obtained responses from 288 adults and 239 children, and we can collect 38 adults and 13 children who developed SARS-CoV-2 infection between November 2020 to January 2021. So the first wave of the COVID-19 pandemic. And what we saw is that the adults had a high rate of obesity and diabetes. You can see here 82% had obesity and 37% had diabetes, but all patients had a favorable outcome. So, and we had a cluster in Andai and they did very well. So, and only 3 children had obesity and none had diabetes, but everybody had a favorable outcome. As far as we know, of course, we cannot be sure that we have all the data of all PWS patients in France. So we conclude that PWS itself is not a risk factor for severe COVID-19, albeit there are high level of comorbidity, and the reason of that is not understood. And as you know me, I have addiction with oxytocin, and perhaps oxytocin levels, particularly high levels in Prader-Willi syndrome, but we wrote that in the paper, can explain parts of the immunity, the differential immunity of PWS versus COVID-19. And to finish the last slide, if we look at the therapeutic perspective, that we should do that, and to target and to control hyperphagia in obesity. I have chosen to discuss the very nice drawing of Andrea Ack paper in Obesity Review published in 2020. And just to focus that it is so hard to obtain a drug that is efficient in hyperphagia. You know, GLP-1 receptor agonist liraglutide, I participate to the study by Novo Nordisk, and the publication or paper were submitted now. So we didn't have a positive effect compared to placebo on BMI, but we have some signal in hyperphagia in adolescents particularly. The potential channel activator DCCR, this drug seems to be good in the hyperphagia, but the FDA requires more data to go ahead on the marketing authorization. The same with the COVID-19 study with the FDA required another study. So it is so difficult to target hyperphagia, and it's really something that we need to develop for helping the development of drug. 2 studies are starting or already starting in the US, but not in Europe. This triple monoamine reuptake inhibitor and the cannabinoid receptor. I know about other drugs that are oncoming. So the hope is still here, but the difficulties have to be bypassed. And we still work on the oxytocin in PWS, and I hope I will have the possibility to give the results when we will have it in this year. So the perspective, what can we learn to improve the care and the understanding from addictive disorders? How to use the new markers IGF-BP7 and the marker of scoliosis like ghrelin. It is a challenge for us for preventing and treating diabetes in the transition period. How to explain the good outcome of COVID-19 and what drug and what targets and for what patients. So for this, I thank you. I hope I'll not be too long, and I'm happy to answer the question.